Dear viewers, greetings. In this video, we are going to see about the classification of antibiotics. Classification of antibiotics. Based on the mode of action, antibiotics are classified into 5 categories. The first category is cell wall synthesis inhibitors. The second category is protein synthesis inhibitors. The third category is nucleic acid synthesis inhibitors. The fourth category is cell membrane alteration antibiotics. And the fifth category is antimetabolites. In the cell wall inhibitors category, 5 groups of antibiotics are present. The first group is penicillins, second group is cephalosporins, the third group is vancomycin, the fourth group is cycloserine, and the fifth group is isoniazid. The antibiotic penicillin group have 5 members. They are penicillin G, penicillin V, methicillin, ampicillin, and carbenicillin. Among the 5 antibiotics, penicillin G, penicillin V, and methicillin have narrow spectrum activity and they are effective against gram-positive bacteria only. And the remaining 2 antibiotics, ampicillin and carbenicillin, are coming under the broad spectrum antibiotic and they are active against both gram positive and gram negative bacteria. Regarding the mode of action, penicillin G, penicillin V, and methicillin deactivate the enzyme that cross the NAM subunit of the peptidoglycan layer of the cell wall. NAM means N-acetyl-muramic acid. And next, regarding the mode of action of ampicillin and carbenicillin, both antibiotics activate the cell wall lytic enzymes and denatures the bacterial cell wall. The second group of cell wall inhibitor category is cephalosporins. The cephalosporins group have 4 members. They are cephalothin, cefoxitin, cefoperazone, and ceftriaxone. All these members have broad spectrum of activity against the gram positive and gram negative bacteria. Regarding the mode of action, cephalosporins group of antibiotics activate the cell wall lytic enzymes and degrades and denatures the cell wall of the bacteria. The third antibiotic of the cell wall inhibitor category is vancomycin. Vancomycin shows narrow spectrum of activity against gram-positive bacteria only, and this vancomycin directly interferes with the formation of alanine-alanine bridges between the NAM subunits and degrades the bacterial cell wall. The fourth antibiotic of the cell wall inhibitors category is cycloserine. Cycloserine is a narrow spectrum antibiotic and it is very effective against gram-positive bacteria. Regarding the mode of action, cycloserine is an analog of alanine that interferes with the formation of alanine-alanine bridges between the NAM subunit of the peptidoglycan and blocks the cell wall synthesis and denatures or degrades the cell wall of the gram-positive bacteria. The fifth antibiotic of the cell wall inhibitors category is isoniazid. Isoniazid exhibits narrow spectrum of activity. Particularly, it is effective against the mycobacterium species like Mycobacterium tuberculosis and Mycobacterium leprae. Regarding the mode of action, isoniazid is an analog of the vitamin nicotinamide and pyridoxine, and it blocks the gene for an enzyme that forms mycolic acids in the Mycobacterium leprae and kills the Mycobacterium species. The next category of antibiotics is protein synthesis inhibitors. Under this category, 5 groups of antibiotics are present. The first group is aminoglycosides, the second group is tetracyclines, the third group is macrolides, the fourth group is chloramphenicol, and the fifth group is lincosamides. The first antibiotic of the protein synthesis inhibitor category is aminoglycosides. Under the aminoglycosides, 8 members of antibiotics are present. They are amikacin, gentamicin, kanamycin, neomycin, paromomycin, spiramycin, streptomycin, and tobramycin. The mode of action or spectrum of activity of the aminoglycosides is broad spectrum activity, and these aminoglycosides are highly effective against gram-positive bacteria, gram-negative bacteria, and mycobacterium species like Mycobacterium leprae, Mycobacterium tuberculosis, and some protozoa. Regarding the mode of action, aminoglycosides inhibit protein synthesis by irreversibly binding to the 30S subunit of the prokaryotic ribosomes, or causes premature release of the ribosome from mRNA and the stopping synthesis. The second antibiotic group of the protein synthesis inhibitor category is tetracyclines. Under the tetracyclines group, 5 members of antibiotics are present. They are deoxycycline, minocycline, tetracycline, oxytetracycline, and chlortetracycline. Tetracyclines exhibit broad spectrum of activity against gram-positive bacteria, gram-negative bacteria, mycoplasma species, rickettsia species, and chlamydia species. Regarding the mode of action, tetracyclines prevent the tRNA molecules which carry amino acids from binding to ribosomes at the 30S subunits docking site. Next, the third antibiotic group of the protein synthesis inhibitor category is macrolides. Under macrolides, 6 antibiotic members are present. They are azithromycin, clarithromycin, erythromycin, natamycin, telithromycin, and clindamycin. Macrolides exhibits broad spectrum of activity against gram-positive bacteria and gram-negative bacteria. Macrolides act by binding to the 50S subunit of the prokaryotic ribosomes and preventing the elongation of the nascent protein and blocks the protein synthesis process. The fourth antibiotic of the protein synthesis inhibitor category is chloramphenicol. Chloramphenicol exhibits broad spectrum of activity against gram-positive and gram-negative bacteria, but it is rarely used for the treatment of typhoid fever. Regarding the mode of action, chloramphenicol binds to the 50S subunit of prokaryotic ribosomes and preventing them from moving along mRNA and blocks the protein synthesis process. The fifth and final antibiotic group of the protein synthesis inhibitor is lincosamides. Lincosamides have clindamycin as its member, and the clindamycin exhibits broad spectrum of activity against gram-positive bacteria, gram-negative bacteria, and some protozoans. Regarding the mode of action, clindamycin binds to 15S ribosomal subunit and stops the protein elongation and interferes with the protein synthesis and completely blocks the protein synthesis process in the bacteria. The third category of antibiotic classification is nucleic acid synthesis inhibitors. Under this category, 4 groups of antibiotics are present. They are fluoroquinolones, niroimidazoles, rifamycin, and clofazimine. The first antibiotic group of the nucleic acid synthesis inhibitor category is fluoroquinolones. Under the group fluoroquinolones, 4 members of antibiotics are present. They are ciprofloxacin, levofloxacin, moxifloxacin, and ofloxacin. And regarding the spectrum of activity, fluoroquinolones exhibit broad spectrum of activity against the gram-positive bacteria and gram-negative bacteria. And in mode of action, fluoroquinolones are the synthetic agents that inhibit DNA gyrase enzyme, which is needed to correctly replicate the bacterial DNA and interferes with the DNA replication and denatures the DNA. The second antibiotic group of the nucleic acid synthesis inhibitor category is nitroimidazoles. Under the nitroimidazoles group, only one member is present, that is metronidazole. This metronidazole exhibits narrow spectrum of activity against the obligate anaerobic bacteria, and in mode of action, this metronidazole attaches with the DNA and prevents its correct replication and interferes with the DNA replication process. The third antibiotic group of the nucleic acid synthesis inhibitor is rifamycin. Under the rifamycin group, 7 members of antibiotic are present. They are rifampin, rifamycin, rifaximin, rimapentine, rimfin, ceticox, and rifaximin. And this rifamycin exhibits narrow spectrum of activity against gram-positive bacteria, mycobacterium species like Mycobacterium tuberculosis and Mycobacterium leprae, Neisseria meningitidis, and Haemophilus influenzae. Regarding the mode of action, the rifamycin group of antibiotics bind to the bacterial RNA polymerase and prevents the transcription of RNA and used with other antimicrobial bacterial drugs. The fourth antibiotic group of nucleic acid synthesis inhibitor is clofazimine. This clofazimine exhibits narrow spectrum of activity against Mycobacterium species, especially Mycobacterium tuberculosis, Mycobacterium leprae, and Mycobacterium ulcerans. Regarding the mode of action, this clofazimine binds to the DNA and prevents the replication and transcription process. The next category of antibiotic classification is cell membrane alteration antibiotics category. Under this category, 4 groups of antibiotics are present. They are gramicidin, niacin, polymyxin, and pyrazinamide. The first antibiotic of the cell membrane alteration antibiotic category is gramicidin. Gramicidin exhibits narrow spectrum of activity against the gram-positive bacteria, and in mode of action, the gramicidin forms pores across the cytoplasmic membrane and allowing single charged cations to cross freely and alters the cell membrane of the bacteria. The second antibiotic group of the cell membrane alteration antibiotic category is niacin. This niacin exhibits broad spectrum of activity against gram-positive bacteria and gram-negative bacteria. And this niacin is a bacteriocin which is produced by the lactic acid bacteria Lactococcus lactis, and it is having a wide number of antimicrobial activity against multiple number of pathogenic bacteria and other organisms. Next, the third antibiotic group of the cell membrane alteration antibiotic category is polymyxin. In the polymyxin group, the antibiotic polymyxin B is the very familiar antibiotic, and it exhibits narrow spectrum of activity against gram-negative bacteria and some amoeba, that is the protozoa. And in mode of action, polymyxin B destroys cytoplasmic membrane of the susceptible cells and results in the cell death. And the fourth and final antibiotic group of the cell membrane alteration antibiotic category is pyrazinamide. This pyrazinamide exhibits narrow spectrum of activity against the Mycobacterium species like Mycobacterium leprae and Mycobacterium tuberculosis, and in mode of action, this pyrazinamide disturbs the cell membrane or membrane transport and prevents Mycobacterium from repairing damaged proteins. By this activity, the cell membrane of the Mycobacterium get altered and results in the death of the cell. The fifth category of the classification of antibiotics is antimetabolites. Under the antimetabolites, 4 groups of antibiotics are present. They are bedaquiline, dapsone, sulfonamides, and trimethoprim. The first antibiotic group of the antimetabolite category is bedaquiline, and this bedaquiline exhibits narrow spectrum of activity against the Mycobacterium species like Mycobacterium tuberculosis and Mycobacterium leprae. Regarding the mode of action, it inhibits ATP synthesis enzymes which are unique to the Mycobacteria species. The second antibiotic group of the antimetabolites category is dapsone. This dapsone exhibits narrow spectrum of activity against Mycobacterium leprae and Mycobacterium tuberculosis, and this dapsone interferes with the synthesis of folic acid in the Mycobacterium species and kills the Mycobacterium species. The third antibiotic group of the antimetabolites category is sulfonamides. Under the sulfonamides group, four members of antibiotics are present. They are sulfadiazine, sulfadoxine, sulfamethoxyazole and sulfanylamide. Regarding the spectrum of activity, sulfonamides exhibits broad spectrum of activity against gram-positive bacteria, gram-negative bacteria, some protozoa and fungi. And in mode of action, sulfonamides binds irreversibly to enzymes that produces dihydrofluoric acid and inhibits the bacterial growth. The final and the fourth antibiotic group of the antimetabolites category is trimethoprim. The trimethoprim exhibits broad spectrum of activity against gram-positive bacteria, gram-negative bacteria, some protozoa and fungi. And regarding the mode of action, trimethoprim blocks second metabolic step in the formation of folic acid, and it results in the death of the bacterial isolates or bacterial pathogens. Dear viewers, that's all about the classification of antibiotics. Thank you for your support. Thank you.